Welcome to the Nanovax podcast, a Nanovaccine Institute curated podcast sharing the latest research and advancements in nanovaccines and nanotherapeutics for human and animal health. The Nanovaccine Institute is a consortium of researchers, institutes, national laboratories, and companies founded and coordinated by Iowa State University.
We are developing nanovaccines and nanotherapeutics for respiratory infections, neural disorders, antimicrobial resistance, cancer, aging, and animal health.
Our vision is that nanovaccines and nanotherapeutics will revolutionize how we prevent and treat disease.
Hello, I'm Kathleen Ross.
I am Balaji Narasimhan.
I'm Mike Roof. This season, you will hear from experts across a range of disciplines who study respiratory diseases and antimicrobial resistance.
The first half of the season will focus on respiratory disease and antimicrobial research within the Nanovaccine Institute. The Nanovaccine Institute is pioneering the development of novel nano-based countermeasures, such as a universal influenza vaccine. We will also talk about other emergent and impactful diseases that have radically changed how we consider and develop policy around global public health.
The second half will focus on the challenges and countermeasures to antimicrobial resistance. Nanovaccine Institute researchers have developed novel nanomedicine therapies to treat chronic disease caused by bacterial pathogens that have developed antimicrobial resistance.
Episodes will explore the different corners of these research topics through presentations on the latest findings as well as conversations among the experts.
Hello everyone, and welcome to this episode. I am Balaji Narasimhan, Director of the Nanovaccine Institute, and Anson Marston, Distinguished Professor and Vlasta Klima Balloon Chair in the Department of Chemical and Biological Engineering at Iowa State University. On today's episode, we will be talking about immunogenetics and the systems biology of vaccine response and performance as related to novel vaccines and adjuvants in both adults and children. And we will be having this conversation with the Vaccine Research Group at the Mayo Clinic. The Vaccine Research Group is in Rochester, Minnesota at the Mayo Clinic, and the group works to improve the health of individuals throughout the world by addressing challenges posed by infectious disease and bioterrorism through clinical, laboratory, and epidemiologic vaccine research. This group was founded in 1989, so they have completed 35 years as we speak as a group together. And they are led by Dr. Gregory A. Poland, MD, who is the founder of the group, and his colleagues are Richard Kennedy, PhD, and Inna Ovsyannikova, also PhD. So Dr. Poland studies the immunogenetics of vaccine response in adults and children. He and his team within the Vaccine Research Group aim to improve the health of individuals across the world by pursuing challenges posed by infectious disease and bioterrorism through clinical, laboratory, and epidemiologic vaccine research. Dr. Kennedy studies the development of immune responses after vaccination. His research focuses primarily on vaccines against viral pathogens, including pox viruses, influenza, measles, and rubella. The goal of these efforts is to understand why some individuals develop robust immunity while others experience adverse events or fail to respond to a vaccine. Dr. Inna Ovsyannikova studies the genetics of innate and adaptive immune responses to both viral and bacterial vaccines. Her interests include vaccine-preventable infectious diseases and the application of mass spec to develop peptide-based vaccines against smallpox, measles, influenza, and bioterrorism agents. So Greg, Rick, and Inna, welcome.
Good to be here.
Great. So since you're celebrating your 35th anniversary, maybe a good place to start would be for you guys to tell us a brief history of how the group was founded.
Well, I came to Mayo Clinic in November of 1988. It's hard to believe it's been that long. And very shortly after that, established the group. This was born of the fact that, one, I was interested in vaccines, had done a lot of study during my own fellowship and residency training. And we were in the midst of a measles outbreak in the US. And the curious observation about this was that many individuals who got measles had received one or more prior doses of a measles containing vaccine. This caught my eye and began to develop hypotheses around why this might be. And that led to a series of grants, including a grant I often kind of chuckle about this, a large grant where we did a family study. Ina was with the group. I don't think Rick was with the group yet. And what was interesting about it is we didn't have computers then. So what we did is we had lists of patients, and we began to realize that people who had gotten the vaccine and were non-responders, by chance, we had enrolled other members of their family, all healthy, and they were seronegative too. Or they had very high levels of antibody response, and other family members also had high levels. So we deduced that this would be genetic in origin, and off we went. And this is now, these kinds of immunogenetic and systems biology type studies that we've done have led to our own characterization of novel vaccines, of understanding immunology and the interaction between immunology and genetics. We're now starting to even look at some social determinants of health and other demographic factors. And it is, we've expanded from measles, which we originally looked at, to rubella, mumps, smallpox vaccine, influenza, and others.
Well, I joined group in 1997, long time ago, as a postdoc in Dr. Poland's lab. And I was so excited about the projects that were going in this group. I came originally from Russia, from Mechnikov Institute of Vaccine and Serum. And to work at the Mayo Clinic in such prestigious lab and learn so much about genetics basis of vaccine response was very, very interesting. And I'm still here with this group and not planning to leave any soon.
Very nice.
And as for me, I'm the babe of the group, I guess. I joined in 2003. And by the time I joined, Greg and Ina had this well-oiled, finely tuned and running machine. And so I jumped in right as they were in the midst of studying some biodefense pathogens and doing some of the same work that we had just discussed with the anthrax vaccine and the smallpox vaccine.
Mm-hmm. So I also should mention that through Greg, Ina, and Rick, the Mayo Clinic and specifically Greg, Ina, and Rick are also part of the Nanovaccine Institute. So we're very pleased to have them participate in our consortium and are looking forward to some collaborative work that we're doing together. So that maybe leads me to my next question is, what are some of the diseases of interest in your work? I know you got started with what you got started with measles, but what are some other things that the vaccine research group at the Mayo Clinic is interested in today?
Well, I'll start and then my colleagues can add in, but we have generally chosen viral pathogens. The exception has been anthrax. We were involved in the largest anthrax vaccine trial that's ever been done. And as a result of that study, by the way, we're able to eliminate one of the otherwise 6 doses that people got. Also defined an unusual side effect called contralateral nodule and symptomatology development. So if you got a vaccine in your left arm and then next time you got it in your right arm, the side effects occurred in the left arm, which is really an unusual manifestation. But for the most part, all of these have been pathogens of immediate public health concern. So we've had outbreaks of measles, we've had outbreaks of mumps. Influenza is an ongoing issue. The concern over both novel orthopox viruses and bioterrorism drove our interest in vaccinia. And again, we got a large amount of funding and did that. Rick and Ina actually developed a very high throughput assay to measure antibody and defined, in fact, a group of individuals that despite having a scar, which is said to be an indicator of lifelong immunity, did not have levels of immunity typically associated with protection. So that was an interesting finding. And we've subsequently expanded that into SARS-CoV-2, avian influenza. What am I leaving out, guys? Anything else?
We've done a little bit of work on Zika and zoster.
Yeah.
I think it started out with sort of the childhood vaccines, the childhood diseases, and morphed into more of the diseases of the older adults. And so now we cover the whole lifespan.
Do you guys study RSV?
We're starting to now, yes.
We are in fact just submitting a grant because it's a particularly interesting time to do so, in that while there is wild virus infection, almost nobody has gotten RSV immunization because the vaccines were just approved a little over a year ago. So it's a really great window of opportunity to study immunogenetics.
Great segue into where we're going next. So let's maybe get an immunogenetics 101 level, sort of beginner level understanding. So what is immunogenetics, and what is the role it plays in vaccine development?
Well, I'm happy to start, or go ahead, Ina, if you want to start, go ahead.
Well, immune response to vaccine depends on many factors, and genetic basis of immunity to vaccination is one of the most important factors. And actually, our group established the field of immunogenetics of vaccine immune response. So we're the first one who studied genetic basis of immune response to measles, to mumps, to rubella vaccine, and showed that genes play a big role in immune response to vaccination, but not only to protective immune response, also there is genetic component of adverse or aberrant response to vaccines. So to better understand how immunity develops and how different factors, including genes, influence of these immune responses, I think extremely important.
Very true. And given the genetic diversity that's present in humans, that is an absolutely big thing to study, especially in animal models. We're not able to capture that. So very happy to see a deep dive into this from the work that all of you are doing.
I'm laughing because if I had to do it over again, I'd have picked simpler questions.
Hard questions are always important to answer, and not everybody can answer them, right? So great to see this work. Rick, anything to add on this?
Just that another element of that, it's not just that genetics can control how well you do or do not respond to a vaccine initially, but it also seemingly controls how long that response lasts. And so another aspect of what we're interested in is it's not good enough to have a vaccine that works for a year or a month. We want vaccines that protect us for decades. And so we're trying to tease out, are there genetic factors that control the durability or the longevity of the immune response?
Very interesting question, and a very important question. So I also mentioned at the beginning of our podcast that your group is using a systems approach to understanding how to build a better vaccine. So maybe we can talk a little bit about what is the systems biology of vaccine development that you all are working on.
Let me start, and then I'll turn it over to Rick to answer the second part. But the reason we moved that way is I had developed this idea of the immune response network theory. So we and others had been, as is typical of science, using quite a reductionist approach rather than a holistic or system approach. And we began to realize that through our work that we could identify at most 5 to 20+% of the inter-individual variability in immune response, measured primarily with antibody, to account for why there were differences. And so we began to think about epigenetics. Now we would add in biologic clocks. we began to look at certain signaling pathways. And so this led to my concept of this immune response network. From there, it was an easy leap into what was then a very nascent field of systems biology. So Rick, if you want to now take that and extend that.
And we recognized pretty quickly that after an infection or after vaccination, the immune response doesn't do just one thing, it does dozens of things. And it's this complicated, intricate, interconnected system. And all of the different moving parts, they have feedback loops, they talk to one another, there's redundancy, there's pleiotropy, it works together as a system that collectively protects us. It's not just one cell making one antibody, or pick your favorite cell doing one effector function. And we said, well, wait a minute, we're really good at drilling down into studying one pathway, one cell, one molecule, but that's not how it behaves. So what if we instead step back and use some of these tools that were coming online recently, where we can look at all of the genes in the genome, the entire transcriptome, the entire metabolome, the entire proteome. What if we step back and use these tools and do a comprehensive assessment? Let's look at everything we can look at and see not just the individual parts, but how they're connected. And so that's really what drove some of our initial work.
That makes a lot of sense. Those kinds of insights are very important to understand, and I'm very glad to hear this approach. You were going to add something?
Yeah, I just wanted to add what Dr. Paul and Dr. Kennedy said, that we're currently doing several systems biology studies. We're doing systems biology study to understand immune response to influenza vaccine in the elderly population. We have grants to study immune systems biology of measles and mumps vaccines. And actually, we are uncovering very interesting molecular signatures that can predict or characterize immune response to these vaccines.
And I should say that Ina is really the one in our lab that has developed a lot of expertise around the influence of HLA genes. That has been really important in our work, because by understanding those differences, by understanding which pathogen-derived peptides, for example, might be promiscuous, we have reversed engineer, and this is where our collaboration with ISU Nanovax Institute comes in, use those to propose novel vaccine candidates using a novel platform approach that's much faster, we believe, will prove to be much faster and better than current, somewhat empiric rather than rationally directed vaccine development.
Yep, makes a lot of sense. And again, I think you're the perfect group to tackle these questions because of your expertise and your approach. So we already mentioned this, but your lab has worked on a large number of diseases. But for the purposes of today's discussion, we're going to try to narrow the focus a little bit and talk about SARS-CoV-2 and flu. And perhaps in a later episode, we can discuss measles, rubella, smallpox, and other important diseases. So maybe we can start with SARS-CoV-2. So can you tell us a little bit about sort of what is the current serosurveillance of antibody titer assessment to coronaviruses in the US population before the outbreak? And how will this research help us understand and inform ongoing responses? We've now lived through several generations of these vaccines as well that have come out.
Yeah, this is really interesting, Balaji, because again, it was an interesting window of opportunity. Certainly, all of us, for sure, by adulthood, we've been repeatedly infected with seasonal coronaviruses, 3 primary ones. But of course, none of us had been infected with what we now call SARS-CoV-2. So it was a completely naive population, which is why it did the damage it did. When I tell this number to people, I think they're so stunned they don't even really grasp it. But as of a month ago, 1 out of every 269 Americans has died of SARS-CoV-2. So we had an opportunity to say, here's a new pathogen, a viral pathogen. we have a growing understanding of how it works. How could we leverage this into understanding and developing a proof case for tandem mass spec technology, where we actually pull off out of the HLA peptide binding groove naturally processed and presented pathogen-derived peptides. So as we say it, we, for the first time in human history, have been able to see what a T cell sees. What are they actually responding to? People have the misconception that they're developing immunity to a virus. Well, in a broad sense, they are. What they're really developing is immunity to a whole spectrum of pathogen-derived peptides. And our pretty simple idea was pluck those peptides off, identify them with tandem mass spec, and then use that in the development of a vaccine. It's, of course, always harder than that simple aspect. And we're learning that there are things we can do, for example, with AI, in fact, with one of your faculty members, where we can enhance the immunogenicity of those peptides by identifying B and T cell epitopes and even attaching various linkers to the ends of the peptides. So that's what we're currently doing, is leveraging that technology into novel candidate vaccine development.
Which I think is very exciting, given where the virus is going, but also with respect to opening the door for new kinds of vaccines.
Exactly.
So just switching to flu a little bit, what are you doing? What is your ongoing research with respect to flu, and what are some particular aspects and findings with your research on influenza that you're particularly excited about?
Rick is just taking the lead in our lab of renewing one of our flu grants. So Rick, you're the perfect one to answer this.
Sure. So with flu, most of our focus has been on older adults. And so with flu vaccination, we know that there's a couple of problems. One is the virus keeps changing. So there's antigenic shift and drift. And so the target keeps moving on us. Second, the people we really need to protect are the older adults. They're more susceptible to severe infection. They're also the least likely to respond because their immune system's breaking down with age, or immunosenescence has set in. And so there's a couple of vaccines available that try and address this. One of them, they simply increased the antigen dose. The other, they added an adjuvant. Both of those work better than the standard dose, but they're not perfect. They're not where we need them to be. And so we recently have finished a study where we took those two vaccines, gave them to older adults, and started looking at differences in not just the antibody response, but also the T cell response. And they're remarkably similar in areas that we think matter for protection. And then we started looking at the transcriptomics and seeing what's going on. We started looking at some high-dimensional flow cytometry. We found differences in innate cell subsets, macrophage subsets, monocyte subsets that we think might help set the stage for the initial scenario where the body sees the antigen, and that's useful for predicting how well or poorly you respond. And we also started looking at senescent cells. We know that senescent cells, they exist, they're metabolically active, but they don't do their job properly. And in fact, because of what they're producing, they can turn other cells senescent. And so now we're embarking on a clinical trial where we're going to use drugs that remove senescent cells, senolytics, and try and use those as an adjunct therapy where we treat individuals before they get a vaccine, then give them the vaccine and see if we can enhance the immune response.
Wonderful. That is certainly an approach that might also have implications for other vaccines against other respiratory viruses that also disproportionately impact older adults, right? So some of the insights you're going to gain from this flu work, I'm sure, are going to be more broadly applicable.
Oh, yeah.
Exciting. So what do you see with respect to, we talked a little bit about formulating these peptides with, for example, novel types of delivery vehicles and nanotechnology. So where do you see that going with respect to the types of things that you're already doing in your lab, and what things do you feel like those would bring to the table?
Well, I think this is really critical. We've been able to bring the science to a certain point, And now what we need to do, and fortunately we do have this collaboration with the Nanovaccine Institute. Frankly, we need people like you and Ratul and others on your faculty who have content expertise we don't possess. And that really is the best science, isn't it? Where there's team science of content experts, where we can literally lateral it over to the next group and produce things that it's like synergy, produce greater than the sum of the individuals involved, because we're able to leverage such expertise. The Nanovaccine Institute has not only content experts, but facilities that would be hard to duplicate anywhere else. So where I think we're going to end, my very optimistic view of this is that we're going to develop a new vaccine platform that will be, I hope, plug and play, so that when so-called pathogen X comes along, we're able to do that. A good base case for this would be H5N1, avian influenza. We're now seeing variants on clades of Mpox. And the current method of developing those vaccines either excludes a large number of people because it's a live virus replicating vaccine, or is difficult to make with a short shelf life, and therefore not amenable, particularly in endemic areas, for example, in the DRC. So I really think this is an exciting era. I mean, the ability to do things at the nano level and build ever more effective and safer vaccines that, as Rick said, would demonstrate durability by particularly stimulating T cell immunity, and perhaps even in the face of immunocompromise or of immunosenescence. If we're able to achieve that vision, we will, in our collective areas, have changed the science, and by that, changed the world for good.
And adding to that is the potential for these types of particle-based vaccines to be stored at room temperature for long periods of time, which is a huge game-changing aspect that enables us to, overcome the logistics of transporting these vaccines to places like the Democratic Republic of Congo or other parts of the world where it is hard to get a cold chain, in place, keep a cold chain in place for long periods of time.
We certainly saw that with the initial release of the mRNA-based SARS-CoV-2 vaccines. They needed an ultra cold, very expensive cold chain. An interesting little piece of history is that I think it was 1, 2, 3 months or so before 2 years ago, the 2022 Clade 2 Mpox outbreak. In the US, we had to destroy, I've forgotten how many hundreds of thousands, or maybe it was a couple million doses of vaccine, because they'd simply reached their outdate by then.
Mm-hmm. Mm-hmm. And that's exactly the kind of heartbreaking thing that we should not be doing.
Yeah.
These are life-saving vaccines, and we want to make sure that they're available when we need them.
Exactly.
So to finish off here, 2 questions. So what do you see as some of the sort of the big challenges moving forward in both your collective research as well as for vaccines? And then what is something that you see as something to be excited or hopeful about in the next 5 to 10 years? So challenges and sort of what's the ray of light?
Let's do this. Let me talk about the policy public health side. Rick and Ina, maybe you can be thinking about some of the challenges that we've faced, and then we can wrap up, as you say, with something hopeful. On the public policy side, we have, in fact, I'm getting ready to be a visiting professor somewhere, and I'm labeling my talk a culture in crisis in terms of vaccines. So, we desperately needed these SARS-CoV-2 vaccines. A mere 1.5 to 2 years later, at best, we get about 15 to maybe 20% uptake of boosters and continue to suffer the consequences of that. So this notion in our society of the politicalization of vaccines has been extraordinarily dangerous and damaging. And it has so separated people that there's not even the opportunity for discussion. In other words, the world becomes a dangerous place when data does not change people's mind and they're resistant to that. And so we have seen now the beginning of outbreaks of measles again because people are not getting their children immunized. I somewhat only half jokingly say, if you want to see polio, well, you have to go to Afghanistan, certain parts of Africa, and oh yeah, New York City. This in the 21st century of the richest, most educated nation in the history of the world. This is even extending into the animal world, which I know is of concern given the profound expertise in veterinary medicine at ISU. We're starting to see rabies and variant rabies viruses because people now bring that vaccine hesitancy to their pets and are saying, well, if I'm not going to get these vaccines, why should I give it to, Fluffy? So that's a place of grave public health and cultural concern for me.
So I'd say another of the challenges we're facing is the fact that a lot of the easy vaccines, so to speak, have been made. Those were for pathogens where it was fairly easy. They were fairly simplistic, fairly uniform. Now we're dealing with things like SARS-CoV-2 or influenza that change a lot. We've got HIV that we've been working for a vaccine for decades. We've got malaria, we've got tuberculosis. We've got these complex pathogens that we haven't, the tried and true approaches haven't worked. So we really need to step back and do something disruptive to change the game so that we can get vaccines for these important pathogens. And Dr. Poland pointed out a little bit about the vaccine hesitancy. Now, part of that's driven by a culture that's very keenly aware of safety issues. And so I think we do need to spend more time and effort on the safety aspect of vaccines.
So going forward, so what is something that each of you are excited or hopeful about looking at the next 5 to 10 years?
Well, for me, number one, I think we will be successful in the development of this platform, and as I say, I think that will be a game changer. Number two is that the science moves forward, regardless of what's happening in the culture and the politics. And number three, the truth always prevails. I'm going to read you a quote from a French philosopher who was also a physician, Marcel Sendrail. He said this was back in 1954. Think how prescient this was. He said, each civilization, by its customs, its laws, its principles of thought, creates for itself a pathology appropriate to itself. He went on to say, a society chooses its diseases and shapes its own pathological destiny. Why is that a hopeful statement, actually? Because the truth does prevail. And as I think wisdom prevails and people understand that these diseases are devastating, unfortunately, it's going to mean kids and others are going to be harmed by vaccine preventable diseases. We will reappropriate wisdom. We will look differently. We will depend on data, recognizing the limitations of science and data, and once again engaged in realistic public health policy.
Well said.
So I'm very excited about age of personalized vaccinology, when we can give the right vaccine at the right dose to the right person. And I think we're getting closer and closer to this to happen.
And I think, as all of you have mentioned, what we need to do is bring together, immunogenetics, systems biology, AI, nanotechnology, policy, all of this together so that we can have the type of impact that we all think is possible, and create the next generation of vaccines for the next generation of society, and help them maybe deal with the pathology of the disease. Going back to the comment you were making, Greg.
For sure.
So this is a great time for people like yourself to be working together and also reaching out and working with others here in the Nanovaccine Institute, and I'm sure with some of your other collaborators. So thank you for the work you're doing.
Thank you.
And thank you for joining us for the discussion today. So other episodes this season will explore other facets of respiratory disease in livestock and humans. And later this season, we'll also talk about antimicrobial resistance, which is another major issue today. So please check out the show notes to learn more about the research discussed in this episode. And please be sure to like and subscribe to the Nanovax podcast in your preferred app or media player.